So let's jump into proteins and membranes. So what we're going to cover is actual structure of proteins. We're going to also discuss the differences between globular and fibrous proteins. We're going to go into a bit of detail on that. And then we're going to look at proteins in their functional context. So basically membranes, membrane transport, and of course, enzymes and inhibition. That's another sort of key thing that proteins, of course, do. And we're going to delve into two specific examples of proteins, namely hemoglobin and antibodies, just because these can be common extended answer questions. Bear in mind that sometimes hemoglobin is also taught in the context of contrasting it to collagen or a fibrous protein. We're not going to cover collagen in a lot of detail here, but just so that bear that in mind when you're doing your case study into proteins. So first part, structure of proteins. The specification points for each exam board are here. I'm not going to go through them, just make them part of your revision. There's more information in there than you think. And let's just jump right into protein structure. So one thing that we need to know about protein structure is the structure of the monomer that make up proteins, which of course are the amino acids. And we need to know the 4 things that are associated with the one central carbon. Oh, sorry. Got to choose the pen. So the structure of the amino acids is something that, yes, you may be asked to draw even from scratch many times. So it's easy. It's relatively easy to learn it. You start with a central carbon atom, which is linked to an amino group here. So that's my amine or amino group, it can be called. A carboxyl group. For those of you taking chemistry, that might be a little bit easier, but even if you're not, don't worry. That's all the amount of chemistry that you'll be asked to memorize, this and a little bit on carbohydrates and lipids. The carboxyl group is what constitutes an organic acid, which is why they're called amino acids, right? The key is in the name. And there we finally have one hydrogen atom here, and we also have the variable R group. And the variable R group is what is going to make the difference between the 20 different amino acids. So we know that there are 20 different amino acids that take part in protein synthesis, and which amino acid specifically you're dealing with is going to be determined by this variable R group. You don't need to know about these structures, the chemical structures of the R group. Just for context, this R group can be anything from a single atom of hydrogen, or it can be a much larger group involving like a little carbon chain. Sometimes if you're not asked to draw the amino acids from scratch, you may be asked to identify whether something is an amino acid. And sometimes they'll try to make those questions hard by showing you a relatively complicated variable group, but you will always look for this pattern here, a central carbon atom with a hydrogen, with an amino group, and with a carboxyl group. What is the primary structure of a protein? This is basically the first step in building a protein in translation. And it is simply a chain of amino acids linked together by peptide bonds. For all biomolecules, proteins, carbohydrates, nucleic acids, the name of the bonds that link the monomer are really important. And they're formed by condensation reactions producing polypeptides. It's important to know that the word polypeptide and the word protein are not mutually exclusive, as we're going to see in a minute. So when you're describing the primary structure, you call it a polypeptide. What is now the secondary structure? It's the folding of the polypeptide chain into alpha helices. So this here is an example of an alpha helix. Pretty easy to recognize because it literally looks like a helix. And beta sheets. Beta sheets will look something like that. So sort of like longer alternating loops. Importantly, and this is something that they will try to sort of catch you with, the secondary structure is held together by hydrogen bonds. Importantly, though, hydrogen bonds that are due to this hydrogen on the basic structure of the amino acid. And we're going to see in a minute why this is important. So keep that detail in your head. What is the tertiary and quaternary structure? The names of the structure obviously imply the stages in making protein, right? So first, the primary structure is assembled at the ribosome with translation. Then the secondary, usually the adoption of the secondary and tertiary structure, just to link it to the parts of the cell where it takes place, they will usually take place inside the lumen of the rough ER. So ribosomes are sitting on the surface of the ER. Polypeptide chain is made. It will enter the lumen of the ER, and that's where the secondary structure and sometimes the tertiary structure as well are going to be adopted. Tertiary structure is, of course, the overall, the final 3D shape of the polypeptide, of the protein. So now the polypeptide is a protein, really, when it has adopted its final shape. So some proteins will stop here at the tertiary structure. They're ready, like most enzymes. So of the polypeptide, now we can call it a protein. We won't call it a protein when it's only in its primary structure, but we can call it a protein now. However, some proteins, some proteins are going to have quaternary structure. And what is, sorry, let me just check that. Yeah. What is quaternary structure? It's when a protein is made up by more than one polypeptide chains. And we're going to see the example of hemoglobin at that. And this is why the term protein and polypeptide are not always mutually interchangeable. So and of course, not all proteins have quaternary structure. And you learn about specific examples, and if you're given an unfamiliar example, it's going to be clearly stated in the question. So our tertiary structure is held together by a list of intermolecular forces that you have to know. And these are hydrophobic interactions is one. What do hydrophobic interactions mean? The variable R group, let me just switch back. This variable R group here can be hydrophilic, meaning it interacts with water. It's polar usually, or it can be hydrophobic. And if it is hydrophobic, it's going to tend to bury itself at the central core of the protein to avoid the aqueous environment of the cytoplasm. So hydrophobic interactions basically mean that you may have two amino acids that have a hydrophobic character in the R group, and they're going to sort of push the protein to fold in a certain way. Ionic bonds is a second one, because the variable R group may have some polar molecules like a hydrogen or a oxygen. Hydrogen bonds, these hydrogen bonds can form between hydrogens and oxygens or another atom between the R groups. So this is where the hydrogen bonds that stabilize the tertiary structure and the secondary structure differ. The ones that stabilize the secondary structure form between these hydrogens and R groups of other amino acids, whereas the hydrogen bonds that stabilize the tertiary structure are between R groups. Finally, disulfide bridges, that's the fourth type of bond that hold together the tertiary structure. And the disulfide bridges are the only covalent bond that stabilizes the tertiary structure. Everything else, ionic bonds, hydrophobic interactions, and hydrogen bonds are ionic interactions. So quaternary structure, as we said, more than one polypeptide. And it can also contain what we call prosthetic groups. A prosthetic group is a non-amino acid, a non-protein chemical group. And we're going to see an example of that in a minute with hemoglobin. Let's look at an example question. Polypeptides are polymers of amino acids. So immediately I can recognize that this is my context here. All of this question is my context. So protein structure, long chains. So these long chains fold into complex shapes that are responsible for many functions within the body. Fine. So that suggests to me if we're talking about long chains, that the focus of the protein, the question, sorry, is going to be on primary structure. My command word is, of course, state. And my direction is, so 1 or 2 word answer. That's what the word state says to me. And the direction, the type of reaction which is required to form bonds between adjacent amino acid residues. So literally naming what is the type of reaction, which is, of course, oops, what happened? Oh, sorry, I'm just going to write it in. Yeah. Which is, of course, a condensation reaction. I'm just going to mention one thing that doesn't pertain only to proteins. The resulting bond from a condensation reaction is going to be different depending on which type of biomolecule you're talking about. If it's a carbohydrate, it's going to be a glycosidic bond. If it's a nucleic acid, it's going to be a phosphodiester bond. If it's a protein, it's going to be a peptide bond. So the name of the resulting bond is specific to the type of molecule that you're looking at, but the name of the reaction is the same, which is a condensation when 2 monomers join up to form a polymer. So here we can see that the hydroxyl group of 1 amino acid, of the carboxyl group here of this amino acid. So here's my 1 amino acid, here's my central carbon, here's the R group, here's the hydrogen, here's the amino group, here's the carboxyl group. Don't always expect to see the structure of the amino acid in a perfect cross like we saw before in the diagram. And here's another amino acid. Here's my central carbon again. And here's the R group, hydrogen, amino, carboxyl. So of course, amino acids react with each other with the carboxyl group of 1 and the amino group of the other. And we have a condensation reaction. Why? Because a molecule of water is released here. So the OH group from the carboxyl and the 1 hydrogen from the amino, they go away and they form a water molecule. Then I now get a direct covalent bond forming between this carbon and this nitrogen here. Often in diagrams, they may ask you to indicate or circle the actual peptide bond. So you'll have to do that correctly, which is this bond here. Let's move on to look at globular proteins. So the description of globular proteins is, I would say, quite a common 3 or 4 marker usually. And so you need to be able to kind of have that in your head. So they are spherical in shape and they are soluble in water. That's a fundamental property of those. How are they soluble? Why are they soluble in water? So this might be an explain question. Because the hydrophobic R groups of the amino acids that have hydrophobic R groups will face in towards the center of the protein. So they'll be somewhere sort of, we just use different colors so that you can see them a bit better. So the hydrophobic amino acids are going to be somewhere in here. They avoid the aqueous environment, which is the cytoplasm. While the hydrophilic R groups are on the outside. So if we kind of, so I'm just going to, so these are my hydrophobic. And these are my, for those that might not know, phobic means that you're afraid of something. So you hide yourself in the interior. Philic means that you have an affinity to something. So my hydrophilic amino acids are going to be on the outside, just all around the outside of my protein. And they mix with water, water being a polar molecule. So my hydrophilic amino acids. I'm going to be abbreviating amino acids as AA. Their shape and the fact that they're soluble in water allow them to be able to, most of the time, be transported along the bloodstream. So they can function as enzymes, they can function as hormones, such as insulin. They can also function as transport proteins, not membrane transport proteins, but general transport proteins like hemoglobin. It's soluble, it can travel across different tissues and organs, and it can travel in the blood circulation, as can hormones, of course. They bind cofactors and many contain prosthetic groups. What is a prosthetic group? So in the case of hemoglobin, the prosthetic group is this little sort of yellow thing that you can see here. Just use different colors so you guys can see that. So this and this will be my prosthetic group. I'm not going to write it in yellow because you won't be able to see. Sorry, let me just change color. Prosthetic group. Prosthetic group is a general term. So it's a small chemical that is not amino acid in nature. In the case of hemoglobin specifically, the prosthetic group is called heme, which is actually where the oxygen binds to. The prosthetic groups in proteins are covalently bonded on the protein. They're a permanent fixture of the protein. And usually we find them in proteins with quaternary structure. What about fibrous proteins? So fibrous proteins have other features about their structure. They've got a regular repetitive sequences usually. They're usually also insoluble in water. So repetitive sequences mean that there might be the same 2 or 3 amino acids repeated regularly. They're insoluble in water in the sense that they may contain amino acids that are hydrophobic in nature. And so they will not easily be dissolvable in water. They will usually have these fibers, and fibrous proteins will almost always have a structural role. So globular proteins, enzymes, hormones, fibrous proteins, structural. Their primary structure is a long unbranched chain. So they will usually not really have a tertiary structure because they don't adopt a circular shape. And this particular diagram is actually collagen, which is pretty much the only example I would say that you need to know from structural proteins. And so we have our long unbranched chain. Sometimes the pen is not cooperating with me. Unbranched chains. And they tightly wind between each other. The fact that they tightly wind gives them more structural support. Often these structural proteins need to have a lot of strength. They provide mechanical strength to the cell or to the tissue or to the organ. Tightly wound. And that forms a sort of rope-like structure. Examples include collagen, elastin, and keratin. Only in, and I think that's more of an AQA question, the structure of collagen is something that you're often asked to describe. And the protein elastin, we're going to see when we cover transport in animals, that is found in the arteries and veins. Keratin is the protein that is responsible for things like nails and these hard structures to form. Let's see an example question. So collagen is a fibrous protein which forms the walls of blood vessels, you said? We're going to see in the structure of blood vessels that they have a layer of collagen and then another layer of elastin. Don't worry about that quite yet. Which forms the walls of the blood vessels, while hemoglobin is a globular protein which travels through the blood vessels. Give 2 ways in which the properties of globular and fibrous proteins differ for 2 marks. So not too long an answer. So what is our again, always breaking down the question. When you've practiced this enough, it's going to become almost second nature. But I would literally go through the motions because you might really see the question in a different light. So what is our context here? This entire first sentence is our context. It tells us which part of the specification, it tells us already that there are going to be structural differences, or rather that collagen is a structural protein, whereas hemoglobin is soluble. And our command word provide 2 reasons, or give 2 ways, really. So here's my command word here, give 2 ways. What is direction? To contrast their structures. The word contrast is not said as a command word in this question. However, it basically means the same thing. How do the proteins differ? So you contrast them. Globular proteins are almost spherical. That's 1 mark. And soluble in water. Sorry, that's the whole sentence is the 1 mark. Soluble in water. While, and often using that kind of comparative language is important, and they look for that in the mark scheme. While fibrous proteins form strong fibers and are insoluble in water. 2 marks. Simple. We are contrasting the properties here, right? So don't be tempted to talk about their function or anything like that. So just one second. I think that the light switched. Sorry, guys, I just realized that I'm going to say the last couple of sentences again. I'd forgotten to turn on the sound. Sorry about that. So I was just going to say that we've got proteins that only reside on one side of the membrane, and we call those peripheral or extrinsic. They'll have hydrophilic surfaces because the cytoplasm and the extracellular fluid are aqueous environments. And we've also got proteins that are completely embedded. They span the membrane, and we'll call those intrinsic or integral. We could also sometimes call them transmembrane. Just be aware of these possible names. And they will have both hydrophilic and hydrophobic regions. So obviously the hydrophobic regions are going to be those amino acids here that are in direct contact with what? With the hydrophobic core, the phospholipid tails, which are hydrophobic. So in order for that protein to actually be able to be embedded in the membrane, they will have to have some hydrophobic amino acids right there. Hydrophobic amino acids. They will also have to have some hydrophilic amino acids, so the ones that are actually pointing outwards to the extracellular fluid or the cytoplasm. What do membrane transport proteins actually do? They span the entire membrane, as we said, and so they'll be known as transmembrane, integral proteins. We've got 2 main types of transport proteins, and one are channel proteins, and they will have a fixed shape, and they will have a water-filled or aqueous, you can say, pore, which allows specific molecules to cross. These specific molecules are most of the time going to be polar. But also, sometimes channel proteins will not allow any polar molecule to pass through. They'll have some specificity about which exact types of molecules they allow. So this, for example, here is showing us an open pore here of a channel protein. And of course, we're going to see in a minute that they are mainly involved in facilitated diffusion. We then also have carrier proteins, and carrier proteins, in terms of function, they can be fairly similar. Sorry, I don't know why this is not pressing. Okay. They won't have a fixed shape. They will have to undergo conformational changes. The reason why this is in bold is because it's typical keyword, and it's a good idea to try to utilize it when making your answers, when structuring your answers. They also move specific molecules across the membrane. Note that carrier proteins may be involved in either facilitated diffusion or active transport, whereas channel proteins are only involved in facilitated diffusion. Let's have a look at a typical exam question. We are shown a diagram. So basically, my context here is this diagram. It's my context. Figure 1 shows a typical cell surface membrane with 4 membrane proteins labeled. Proteins B and D transport substances across cell membranes. Further illustrating the context here. We're talking about transmembrane proteins. Identify proteins. This is my command word here, of course. Sorry, pardon me 1 second. Something seems to have disconnected. I'll be back in 1 second. Let me just quickly sort that out. Okay, sorry about that interruption, just a small technical problem. So we said this is the entire diagram, and this description is our context, and we are, sorry, just change color. So our command word here, of course, is going to be identify and state whether they are extrinsic or intrinsic proteins. So that'll be my direction here. And it's only a 2 marker, so that's going to be a relatively straightforward answer. So first of all, I'm looking at their structure. I'm looking at B. I'm looking at D. I can see that they span the entire membrane. So my answer, name, command, name, So my answer is going to be that they are, that they are both, or I can just start my answer by identifying them actually. So B is a channel protein. How do I know that? How do I know that it's a channel and not a carrier protein? Because I can see that it has this kind of lumen or pore here. And I can see that this is kind of like just open. Whereas if I look at my protein D, I will call that is my carrier protein. And how do I know that? Because I can see that part of my protein down here is sort of closed and it's open towards the outside. So typically what this carrier protein is going to do is a substance that needs to be transported is going to bind up here to the open side. The protein will then undergo a conformational change. It will open the other way, basically, and then the substance can be released into the inside of the cell. So 1 mark goes for identifying them. B is channel, correctly identifying them, of course, right? B is channel, D is carrier protein. That's my 1 mark. And then both are intrinsic proteins because they span the membrane. We don't need to justify why, I'm just telling you. Proteins. Of course, here I'm calling them intrinsic and not integral or transmembrane because that's the phrasing that I'm being given in the question here. Let's then go to actually look at how transport across membranes actually works and what are the types of transport that we can have. We can have passive or simple, it's often called. Simple diffusion. The word diffusion is always passive in the sense that it doesn't require energy, sorry, not enzymes. Passive or simple diffusion is used for small and non-polar molecules that can diffuse down their concentration gradients through the membrane. And they have to be non-polar or hydrophobic because they have to easily, as we said, be able to cross the hydrophobic core of the phospholipid tails in the cell membrane. If you don't remember exactly why phospholipid tails are hydrophobic, definitely revise that part of your syllabus quite well, your specification quite well, because it's really key to be able to understand the reasons why different molecules move into the membrane, through the membranes in different ways. We then have facilitated diffusion, which is diffusion again, right? So it's diffusion, so no ATP required. And again, molecules move down their concentration gradient. If it's diffusion, they're moving down their concentration gradients and they don't need energy. However, facilitated diffusion will utilize our channel or our carrier proteins and will allow slightly larger, it's not going to be an entire protein, but it can be an amino acid, for example. And polar, crucially, molecules or charged molecules, such as, typical example of charged molecules, sodium or potassium ions. For those of you that have already done the year 13 content, you'll know that this is really used in neuronal communication, nerve impulses. So finally, we have active transport, which of course, as the name suggests, requires energy in the form of ATP. It uses carrier only, not channel proteins. And it uses energy that's derived from ATP, or more specifically from the hydrolysis of ATP. Super quick reminder, ATP switches between, ATP switches between the states of ATP, where it three phosphates, and ADP, which has 2 phosphates. And it can move molecules against their concentration gradient. Super important qualitative difference between the 2 types of diffusion and active transport. And there are specific examples that it's good to remember for active transport. In plants, it's the uptake of minerals from the soil, if you want to make a quick note of that. Uptake of minerals from the soil, from the root hair cells. In the intestine, the absorption and uptake of glucose and amino acids. Let's see a typical question. Small nonpolar molecules move through the plasma membrane via simple diffusion. So This is my context. And it's also something that I'm not going to repeat in my answer, as we said before, from the examiner's reports. Outline the different ways. Outline is, of course, here my command words. The different ways that charged, polar, and larger molecules move through the membranes compared to small, non-polar molecules. Marks were rewarded for quality of written communication. This is what I said, that sometimes it's explicit. It basically means that don't make your answer in bullet points like the mark scheme. Actually write full coherent sentences. So my direction here, so my context is the movement of membrane of molecules through the membrane. My command is to describe, basically outline or describe. The only challenging thing I would say is that sometimes the word outline might actually mean describe, but always refer back to the exam technique session that we did in the beginning. And the direction, you focus on the 3 types of transport, sorry, I should say here, not molecules. Transport. Well, the 3 types of molecules that move with the 3 different types of transport. And this is, of course, a 5 marker. So there's a little bit of information that we have to say here. So charged, polar, and larger molecules cannot use simple diffusion to cross the membranes. That is 1 mark. And again, when we say larger here, we don't mean proteins. Proteins move by an entirely different way, endocytosis. Larger substances are transported using carrier proteins. That's another mark. You could have also said endocytosis here if you wanted. That would have been acceptable here. Over endocytosis. The process may use active transport. These processes may use active transport, moving substances against their concentration gradients. That is 1 mark, mentioning that they're going against the concentration gradient, or also use facilitated diffusion down the concentration gradients. That is another mark. Polar and charged substances diffuse through channel proteins. That is a mark. By facilitated diffusion, or are transported by carrier proteins. By some mark. By either facilitated diffusion or active transport. Sorry about that. That's another mark here. Don't forget that in the mark that you're awarded for clear phrasing, spelling is also checked. So worth checking that you always have the right type spelling. The enzymes is another class of membrane proteins that we can have. Of course, we know the definition of an enzyme. They act as a biological catalyst by reducing activation energy. The induced fit model is today the sort of most widely accepted model for how enzymes work. They don't have a fixed shape that is exactly complementary to the substrate. Rather, it's the proximity of the substrate to the enzyme that results in a slight change to the shape of the active site to make it complementary. The use of the word complementary is always usually underlined in mark schemes when you're describing any kind of enzyme questions. This allows the active site to mold itself to the substrate, and it forms a stable enzyme substrate complex. How does enzyme inhibition work? We've got 2 main classes, 2 types of inhibitors. One is a competitive inhibitor, or a non-competitive inhibitor. And they work in 2 distinct ways. And the naming here is quite telling of what they do and how they work. Competitive inhibitors bind to the actual active site. So what does that mean? It means that they will have a similar shape often to the substrate, right? Because otherwise, how could they bind to the active site if they don't have a at least partly complementary shape to the active site? So they physically block, they physically compete with the substrate for the active site. And so that makes a difference to whether they are affected or not by substrate concentration, because we can imagine that if we massively increase the concentration of the substrate, it will outcompete the competitive inhibitor for the binding. If there are more substrate molecules around, there's more chances for the substrate to collide with the enzyme and create a enzyme substrate complex than the competitive inhibitor, which is in smaller amounts. Non-competitive inhibitors, on the other hand, do not bind to the active site. They bind to an entirely different site of the protein, and they cause what we call an allosteric change. So by binding, as you can see here in this diagram, they will indirectly change the shape of the active site such that it no longer is complementary to the substrate. And so they will be unaffected by the substrate concentration. You can add all the substrate that you want. If the active site doesn't have the correct shape to bind the substrate, you've got no reaction. Let's see how that difference looks like. So these are quite sort of common graphs that you might be asked to interpret. So we've got our rate of our reaction on the y-axis and substrate concentration. This means that basically scientists will perform the experiment at a given substrate concentration, measure the rate. They'll then perform the experiment again at a higher substrate concentration and then measure the rate. And this is how these curves are constructed. And we'll often be asked to basically identify the shapes of these curves and deduce whether they come from a competitive inhibitor, an uninhibited reaction, which is the purple line here. So this is the uninhibited normal, I guess you can say, uninhibited reaction. And as I expect, my uninhibited reaction, as I increase the substrate concentration, so will the rate of the reaction increase until it reaches a plateau. What does the reaching of plateau mean? Something else is now the limiting factor, the amount of enzyme. So all of the enzyme active sites have been occupied here. So adding more substrate will have no effect. Just bear that in mind, because sometimes you might be asked to sort of explain the shape of this curve a little bit further in longer questions. In the green color here, we have our competitive inhibitor. And why do we have that shape curve? Because the adding more substrate will increase the rate of the reaction. So the reaction can eventually reach the same rate as the uninhibited reaction. So solutions containing the competitive inhibitors are able to reach the uninhibited maximum rate of a reaction with a high enough substrate concentration. I'd encourage you to use that phrasing if you want in your answers. Meaning that we can basically effectively drown out the effect of the inhibitor by adding enough substrate. However, if we are using a non-competitive inhibitor, like in this curve, then the reaction will not reach the uninhibited maximum rate of reaction because the active sites are themselves not able to bind the substrate properly. So adding more substrate is not going to help the maximum rate of the reaction. Let's see an example question. It's a relatively short one. Sometimes they can be a bit longer. Explain. So, of course, my command word here is explain. Sorry. Okay. Explain why non-competitive inhibitors are unaffected by substrate concentration. By context here is that substrate concentration and non-competitive inhibition, the relationship. My command is to give reasons, to explain, and my direction is to only focus on non-competitive inhibition. I don't need to explain anything about competitive inhibitors here. Non-competitive inhibitors bind to allosteric sites. Sorry, let me just. Something peculiar is happening with the pen, so just give me a sec. Non-competitive inhibitors will bind to allosteric sites on the enzyme. That is one mark, just stating where they bind. So they don't need to compete with the substrate for the active site. That is my second mark here. We're going to look at some examples of proteins. And we are going to start by hemoglobin. So this is, again, specification points. And this is like our case study into specific types of proteins. Hemoglobin is an example of a protein with quaternary structure. So it contains more than one polypeptide chains, namely 4. 2 of them are alpha chains and 2 of them are beta chains. It is, of course, a globular. It has a roughly spherical shape. And it is conjugated to the prosthetic group that we said is called heme. Sorry, my pen appears to not be cooperating. So just give me just one quick second. I do want to be able to jot things down for you guys. That's peculiar. Sorry about that. I'll sort it out before. I'll move on now because we need to get through this, and I'll sort it out for next session. Oh, it appears to be working somewhat. It's a globular protein, and it's conjugated to what we call the prosthetic group, which is called heme. So this here is the structure of heme. You do not need to memorize it. You do, though, need to remember that it contains iron, and it's crucial. Iron is crucial for the function of heme. Each heme group can bind to one oxygen molecule. And since there are 4 heme groups, 1 on each polypeptide chain, each hemoglobin molecule can bind 4 oxygen molecules in total. Again, for those of you not taking chemistry, 1 molecule and atom of oxygen are not the same. Sometimes they try to catch you with that. 1 molecule of oxygen contains 2 atoms. So you've got 4 molecules, but 8 atoms in total. Just remember that because they do try to catch you sometimes. It's important that we are able to describe and explain the hemoglobin saturation curve. So hemoglobin is found inside red blood cells, and it will basically transport oxygen from the lungs to the respiring tissues to allow for aerobic respiration, of course. Respiring tissues are basically all of our tissues. There's no alive cell or tissue that doesn't respire, that doesn't need oxygen. Hemoglobin undergoes conformational changes when it binds oxygen, and these conformational changes are collectively known as cooperative binding. What does cooperative binding actually mean? And sometimes we're asked to define what cooperative binding means. It means that and again, I'd advise you to sort of copy that phrasing for consistency. Binding of the first oxygen molecule changes the orientation of the heme groups, or if you don't remember that, you can just simply say that it causes a conformational change to hemoglobin such that it makes it more accessible to more oxygen molecules. So what it means is that how do we connect that to this curve? By the way, this is not required for Edexcel A. It means that when we have low partial pressures of oxygen, the word partial pressure is just another word for concentration, but specifically for gases. It means that the first oxygen molecule is going to bind slower. So it's going to bind slower, and it's going to, however, cause a conformational change such that the second and third oxygen molecules will bind more easily. So at these middle partial pressures here in this middle range, this sort of like steep range of my curve is demonstrating that the second and third oxygen are binding quite efficiently. And at high partial pressures of oxygen in that final bit of my curve, is where my hemoglobin is actually going to get completely saturated with oxygen. Let's have a look at a classic exemplar question. Red blood cells are crucial for the transport of oxygen to the tissues in mammals. Many molecules of hemoglobin are present within each red blood cells and contribute to their function. Describe the role of hemoglobin in oxygen transport in mammals. 3 marks only describe, no explain. What is my context? Hemoglobin in red blood cells and the fact that it's important for cell function. What is my command? To write out the role of hemoglobin. No. Why? And what is my direction? Only focusing on the oxygen transport functions of hemoglobin. So, hemoglobin has a high affinity for oxygen when the oxygen partial pressure is high, meaning what? That when hemoglobin is at the lungs, it will bind regions of high partial pressure of oxygen. So where? Let me just show you. Let me just relate it to the curve. So the top right, sorry, I can't jot down here, but the top right-hand bit of the curve with high partial pressures of oxygen, that is where hemoglobin is going to have a high affinity for oxygen, and it forms oxyhemoglobin. So that's 1 mark. It then transports the bound oxygen to respiring tissues where the oxygen is released. Why? How? Because oxygen is released at the low oxygen partial pressure, so the bottom left part of my curve, at respiring tissues. Respiring tissues have low partial pressures of oxygen because they're using the oxygen, of course, for respiration. And so my hemoglobin affinity is going to have a lower affinity at the respiring tissues with lower partial pressures. So the key here in describing the role of hemoglobin is describing the idea that hemoglobin has different affinities for oxygen depending on the partial pressures of oxygen. High affinity when oxygen is abundant, high partial pressures. That's good because it can bind oxygen. Low affinity at low partial pressures. Let's look at antibody structure. Antibodies are another classic type of proteins that we have to know about. So antibodies are also examples of proteins with quaternary structure because they're also made by more than 1 polypeptide chains, 2 light, 2 heavy. The heavy chains, it's unfortunate that I can't label here. The heavy chains are the long ones, so the dark blue ones that you can see. And the light chains are the smaller green ones on each side here. They're called light and heavy because if you centrifuge them, the longer chains will sink further down to the bottom of the tube. So the terms light and heavy only refer to their size and nothing to do with their function. We've got 3 general regions that we have to know about in the antibody. One is the variable region. So that will be the light blue and the light green. And they contain antigen binding sites. And it makes sense that they are, so I just figured out that I can actually use my finger. So this here is my variable region. And they have a unique amino acid sequence, which means that they will have a specific shape and they will recognize specific antigens. So the variable regions, as the word suggests, are specific to the antigens that antibody is going to bind. We then have the hinge region, which is sort of this little region here. Sometimes they'll show you the little disulfide bridges that the hinge region is made of. And we also finally have the constant region. So this and this is the constant region. So the variable and the constant regions can be on both the light and the heavy chains. Make a note of that. And of course, as we said, the antigen binding sites are specific to individual antigens. What do they do? What do antibodies do? They don't actually directly destroy the pathogen. They rather mark it for the antigen to be, or the pathogen, the entire pathogen, to be destroyed by other cells of the immune system, such as the phagocytes, neutrophils and macrophages. They cause what we call agglutination. And the word agglutination was actually in your little table of important words and prefixes. Agglutination basically means clumping together. So as we can see, as we can see, there are in the hinge region, so the hinge region down here, it allows each antibody to bind two different pathogens because we've got two variable regions per antibody molecule. So one single antibody molecule can bind more than one antigen. The variable region actually binds to the antigen itself. The antigen might be a structural protein on the surface of the pathogen itself, or it may be a toxin that the pathogen secretes. And the constant region actually is a binding site for the phagocyte. So as we said, the antibodies basically mark the pathogen for destruction as a marker for phagocytosis. So the phagocytes are actually going to bind here on the constant region of the antibody. Let's have a look at a typical question for antibodies. So compare antibodies with hemoglobin to discuss the relationship between structure and function in proteins. Now let's really break this down, the 6-marker down. My context is protein structure and function. Okay, that's relatively obvious. My command is to compare both the structure and the function. So, and here I'm going to actually, because it says compare and not just contrast, I will use both similarities and differences. And what is my direction? To explain how structure and function are related, which is a really common type of question as well. Let's look at the answer. Both hemoglobin and antibodies are globular proteins, a similarity, 1 mark, with 4 polypeptide chains, which are soluble in polar solvents, as most globular proteins are, another mark. They use their solubility to travel around the body in the blood, similarity. Similarity in terms of function, right? One was similarity in terms of structure. This is similarity in terms of function. They can both travel around the blood. But antibodies travel to the source of infection while hemoglobin transports oxygen and carbon dioxide. Difference in function. 1 mark each. Antibodies will have a different variable region, which allows them to bind to a variety of antigens, while the structure of hemoglobin is constant between molecules, and it contains 4 heme groups, which allow hemoglobin to bind oxygen. So contrasting here now the structure as well. Let's have, sorry, a sec. Yeah. Because this is a pretty typical question, comparing different types of proteins, particularly antibodies or hemoglobin, or in instances, hemoglobin against collagen, which is also fibrous. All of the points that you want to make are kind of summarized in that in this table. And I find this a super useful practice for preparing for potential extended response questions. So you're comparing the structure here. The primary structure is variable, whereas the primary structure is constant. I'm not going to go through this in a lot of detail. They're globular, soluble, but as a similarity, not a conjugated protein, whereas hemoglobin is a conjugated protein. A number of structural differences, and also a summary of the functional differences. Again, I'm not going to go through these in a lot of detail. They're basically what we covered in that question, but feel free to familiarize yourself a lot and pay note to the phrasing and the use of keywords in bold